the Lustgarten Foundation and Stand Up to Cancer, started something called the Pancreatic Cancer Collective to really push the envelope much harder than we have in the past. We are so excited to welcome Drs. Martin McMahon and Conan Kinsley to our community.
For many years we have understood that pancreas cancer is a disease that is driven by the aberrant activity of a protein called KRAS. And KRAS has been a very difficult protein to target pharmacologically. There are no drugs currently that target the driver protein in pancreas cancer. KRAS sends signals throughout the cell through the activation of a number of downstream signal transduction pathways. And one of these is a pathway that we've spent many years studying. Dr. Eric Collison, who is a fellow in my lab, showed that is the key pathway for pancreatic tumorigenesis. Now we can see killing of cells in a petri dish, and we can see shrinkage of tumors in animal models.
Dr. McMahon published a new method to treat pancreatic cancer where he mixed two medicines. One, a medicine you would take if you had bad malaria. Second, a medicine you would take if you had melanoma. So these two medicines, when mixed together, forced the cancer cells into a situation where they died.
There was a patient who had received many chemotherapies, had also received surgery for pancreas cancer, and sadly the disease had continued to progress. And so he was really at the end of his options in terms of chemotherapy to treat the disease. And so Conan asked him if he was interested in the possibility of trying this experimental therapy, which is a combination of two drugs. One drug is called trametinib, and the other drug is called hydroxychloroquine, one to block the MAP kinase pathway, one to block this process of autophagy. And our patient, who was really quite sick, actually got an enormous response to this combination therapy. His biomarker in the blood, that is a measure of pancreas cancer progression, declined from over 33,000 to less than 1,000 in about 2 months. But even more importantly, CT analysis of scanning of him showed that tumors that he had in the liver that were making him quite sick actually went away very substantially. And so we were able to go from the petri dish to a patient in the space of just over 2 years. This was the foundational work that we were able to present to the Pancreas Cancer Collective and to tell them about this exciting new series of developments.
My hope for Dr. McMahon's work is that he will enroll many patients on this trial. We will learn a lot about how to best administer these medicines such that we can maximize the benefit to the most patients.